Good afternoon, everybody. Welcome to this plenary lecture. This is the Hypothalamic Neuroscience and Neuroendocrinology Australasia plenary lecture, and it's my pleasure to introduce the talk today. We call this HNNA, so if you hear that, HNNA is the term we use. Firstly, my name's Zane Andrews. I'm the current president of HNNA, and it's my pleasure today to introduce our plenary lecturer, Professor Dave Grattan. Otherwise known as the Prolactin guy on Twitter. So I think you should follow him. So actually I went to test this, and I went on ExpertScape to have a look, and he's actually ranked #1 out of over 13,000 people that have published on prolactin. So I think he is actually the Prolactin guy. It's a testament to Dave, this reputation of prolactin, and it goes all the way back to his first paper in 1989, where he actually, by the looks of the title, failed to create an ELISA to measure prolactin in the blood. And I just it said failed on the title. I just looked recently, he had, according to Scopus, his 200th paper, and it was still on prolactin secretion. So I think that's a real testament to Dave, his tenacity. He has stayed focused on understanding prolactin and how it works to particularly regulate pregnancy and the effects on the maternal brain. The other thing I should say about Dave is he's always very, very generous with his time, as I'm sure many of you people would have experienced over the years. For example, he's been the editor-in-chief of Journal of Neuroendocrinology. He's the current director of the Centre of Neuroendocrinology. He's former head of department in Otago. He's always giving his time, which explains why it takes 5 days to get an email response from him. He's also been very proactive in promoting research. He's been a big part of the community to promote neuroendocrine research. And he's also promoted ECRs. And I think this is where I came in. So Dave was actually my PhD supervisor many years ago. And I must admit, I was in the third year anatomy class, and I wanted to do honours, and I didn't have a burning desire to understand prolactin secretion, but I knew I wanted to do my research with Dave because he had a lot of passion for the research, and you know, he stood up and he made it fun. So I'm going to take the prerogative to just show you a couple of slides of the time. This is 20 years ago at the ICN meeting in Bristol, and that's after the conference dinner. And on the way home in the bus. Now, many of you would have experienced having a few drinks with Dave. He likes to have fun, and as you can see here, he's pretty good at dancing as well. This is on the dance floor at that very conference. But yeah, so Dave for us made research fun, and there was always the celebration of research achievements within the department. And I don't even know what that is. It's just an old photo I found. He also liked donuts, nothing more. And if you go to a conference with Dave, you will inevitably find a sporting event, whether it's cricket, rugby, or baseball. And so I just wanted to take this final slide. Essentially, you know, here we are doing some research, I guess, but passing on the passion for research. And I think that's what really stands out for me. So thanks, Dave. It's a pleasure to introduce this talk. And yeah, we look forward to it.
Thank you, Zane, for that really kind introduction, and thank you very much to the organisers for giving me the opportunity to present this plenary lecture. I've actually been really excited about this for weeks, but that's been bad for me because I've rewritten this talk about 15 times, and I'm going to give you one version of them, and hopefully it's the right one. And I feel like I'm going to need to use a pointer, so we do have a mouse here, so I probably will be able to do it at times. Okay, so in the program, my talk was advertised as prolactin and behaviour, and I am going to cover aspects of prolactin and behaviour, but I think the point I want to make out of the whole of my talk is that I see prolactin having a really wide role in pregnancy and lactation, in coordinating physiology, behaviour, so that the mother can deal with all of the changes that are happening in her body, in her interactions with the baby, etc. So I've kind of renamed it Prolactin Pregnancy in the Preparation for Parenting, and I will get into it, I guess. So the plan is to give you a few thoughts about prolactin. You guys probably all have a pre-existing opinion about what you think prolactin does and what it's there for. I'm going to try and change that opinion. And then I'm going to talk about this idea that prolactin is involved in this adaptive response to pregnancy and lactation. And I'll give you 4 sort of research anecdotes. There's not enough time, obviously, to go through a whole lot of scientific detail, but I want to show you some of the actual experiments that are happening in the lab at the moment, and hopefully quite a bit of unpublished data. And then we'll finish by sort of pulling it all together again and thinking again about how we view this role of prolactin. So what's in a name? I found this picture of the cover of Time magazine from 1939, and that's a picture of Oscar Riddell, who was recognized for his pioneering work purifying and naming the hormone prolactin. It's the hormone that was involved in milk production, so prolactation. It's a great name. No doubt there are some of my French colleagues in the audience who will be thinking, oh yeah, but some French guys discovered that like 2 years earlier than he did. But anyway, Oscar's name stuck, prolactin. And I think it is basically, it's pigeonholed the hormone around its major function to control lactation, milk production and lactation. And I would say if I polled the room, that'd be the first thing you wrote down if I said, well, what does prolactin do? Milk production and lactation. And what I've been thinking about is why that perception is probably wrong. So the first thing, this is some data that is a relatively new Cree line that we developed to map the distribution of the prolactin receptor. There's been many studies over the years that have mapped the prolactin receptor using different methods. And the key thing is it's found in all sorts of places in the body. Shown here, we've got the brain, the pancreas, it's in the beta cells of the islets, it's in the epithelial tissue in the kidney, in the gut. It's in a range of other places that are not mammary glands. So when you look at it, something like this for a hormone, its distribution all over the place, it's clearly doing other things in addition to lactation. This is another way I've sort of got to think about it. If you think about the whole of the vertebrate life, and you think of all of those, how many of them have prolactin? It's pretty much all of them. And you think how many of those lactate? We're just talking about the mammals. And in fact, even within the mammals, it's only the females. So you would kind of look at that and think, well, what's the function of the hormone prolactin given this context? It seems unlikely that evolution is going to have put together a hormone, let it sitting around for 250 million years until the mammals showed up, and then allow half of the mammals to use it for lactation. It's probably doing something else. And I'd like to talk to you about what else it might be doing. And my interest in the what else actually started a long time ago when I was the only thing that I was set out to study at that time, back when Zane was a PhD student and I still had hair and slightly smaller trousers. Back in those days, I was interested in the control of prolactin secretion because I was interested in how did prolactin control lactation. And the question I had was, well, how does prolactin get high in lactation if it's always, it's got a negative feedback loop that should be turning it off? And I started thinking, well, maybe there's a downregulation of prolactin receptors on the dopamine cells in the brain. And at that time, there really hadn't been a description of those dopamine receptors, the prolactin receptors on the dopamine cells in the brain. So I went looking for them. And so, just to orient you, if I can get this mouse working, right down in the bottom corner of the slide, for those of you who are in the front, you'll be able to see where the arcuate nucleus is. This is where I was looking for prolactin receptors, about here. But as soon as we started looking at the brains and staining it in various ways for prolactin receptor, we saw that it was all sorts of other places. And this slide is, again, this is now our prolactin receptor Cree line. It's been labeled with a reporter for tau GFP, which labels the cell bodies containing prolactin receptors and their projections. But what you can start to see is that prolactin receptors are widely distributed in the brain. And most of you will have your favorite brain area, and you can pick it out on the slide. And unless you're Lisa Galea, and that's the hippocampus, you'll find that there are probably prolactin receptors there. And so you can start to think, well, why would prolactin be doing that? Another piece of data that actually I have just generated in the past week or so is I bumped into Giles at a conference, as you do, and he was presenting some information on his new project, which is something that Brian Lamb and himself are calling the hypomap, where they have put together sort of a whole series of single-cell sequencing studies in the public domain into a single database, and it allows you to interrogate it. And I said to Giles over a glass of wine or something, hey, could we actually use that and find out how many cells have the prolactin receptor? And Giles, of course, said, sure, Brian will do that for you. And so Brian did that for me. And so this is a little piece of data that's very, very a rough overview of all of the cells that they've identified in the database. The neurons are largely in this cluster, and you can kind of see the big pink blob are largely glutamate cells. The orangey, whatever color that is, are the GABA cells. And within that, they've identified about 130 specific neuronal clusters. So how many of those are going to have the prolactin receptor in them? Well, this is what we found. Well, this is what Brian was able to show me. Basically, in about 15% of all of the neurons that they interrogated in the database expressed a significant amount of the prolactin receptor. And if you looked at those individual clusters, over half of those clusters, more than 10% of the neurons within that cluster expressed the prolactin receptor at a significant, at a significant quantity. And so you can actually probably see here, there's a couple of hot spots. It's the Kindi neurons, and the dopamine neurons, and that's kind of reassuring because those are the two that I've published on saying that they've got prolactin receptors in them. So that's nice. But there's actually prolactin receptors in a whole lot of other places. So what is that all about? Hopefully you're now thinking, why the hell would there be so many prolactin responsive neurons? Because that's exactly what I was thinking. Why, if prolactin is just a lactation hormone, would so many different neurons have the prolactin receptor and so many different tissues? And so that's really what I'm going to try and take you through my ideas, the way that I was thinking about this question, and then we'll show you some actual research data to take you through that. So the first thing I started thinking about, well, when is prolactin high? And obviously prolactin is high in lactation. When else is it high? What sort of functions might need to change in the body when prolactin is high? Because that might give you a clue as to why would a particular tissue have prolactin receptors. Is there any evidence that prolactin might affect that function? And it's this question why many of you will have met me in conferences before, and you'll find me kind of annoying because you'll be describing some wonderful bit of physiology, and I'll put my hand up and say, well, how does prolactin affect that? And the reason genuinely is because there's prolactin receptors there, and I'm just trying to understand why would that need to change. And sometimes people have thought about it, mostly not, and we can have a discussion. It's some good advice for the students, the trainees. Actually, I was talking to Lisa after her talk, and I thought, trainees, it's always nerve-wracking asking a question. So here's a couple of questions for you if you're at a neuroscience conference. One from Lisa's talk, right? Pretty much every talk, you stand up at the end and say, why didn't you study that in females? Okay, and from my talk, what you'll be able to do is, almost every single talk, you'll be able to stand up and say, how does prolactin affect that? No, there you go, 2. It's not stressful at all. And the final thing that I was sort of thinking is, how is prolactin going to influence these functions? Is it one-on-one, one cell has a prolactin receptor, that's how it happens? Or is it a more dispersed network type of level of activity? So the sorts of things I've been thinking about is, when is prolactin high? But it's obviously high in lactation, but actually it's also high in pregnancy, and this is true in many, many species. This is just data from women, so from human pregnancy, and I've just redrawn this data from an absolutely awesome proteomics study that essentially took blood samples throughout pregnancy in women and did proteomics to work out all of the proteins that they could detect. And of 1,300 proteins that they could detect in blood, the top two of the top 1%, so 2 out of the top 10 basically, were prolactin and human placental lactogen. So these 2 hormones are massively increasing in pregnancy. Now you could think that that's all about lactation, but remember all those other cells that have got the prolactin receptor, they're seeing it too. So I actually now think pregnancy is the key time when prolactin is having its action. It certainly carries on into lactation, but I've been really interested in what is it doing in pregnancy. And then what systems might need to change when prolactin is high? Well, when prolactin is high, we're talking pregnancy and lactation, and almost everything is changing in the woman, in her body during that time. And this was a figure I put together for the latest edition of the Physiology of Reproduction book. And it was nice to see Tony somewhere in the audience. He was the editor of this version. And you can see that there's a bunch of things happening in the body. I'm not going to go through these in any detail. We'll come back to the slide a few times. But lots and lots of things happening in the brain. And so, I was interested in maybe prolactin was acting here. Certainly, at least some of the adaptations that happen in pregnancy and lactation are dependent on prolactin. And this is a diagram that was put together by Bob Bridges, describing the network that controls maternal behavior. And you can see that, just not sure that I've advanced the slides right. You can see that the middle of this network is an area called the preoptic area, medial preoptic area, MPOA. And Bob has identified that prolactin here is influencing maternal behavior, acting at this spot. And that's from work that he did many years ago, injecting prolactin into different parts of the brain in rats, and seeing that but when he injected it into the preoptic area, he could induce maternal behavior or advance it. A few years ago, Rosie Brown, when she was working with me, knocked out prolactin receptors specifically from that part of the brain. And we found that that was catastrophic for maternal behavior. So basically, if we deleted the prolactin receptor from the preoptic area, the mothers would give birth, apparently healthy babies. She would sort of clean them up and do everything that looked like normal maternal behavior, but then she would kind of get sick of it and push them out of the nest and abandon them and would never look after them. So she would just not look after the babies in the absence of this signal in the brain. So prolactin action, as we said here, is necessary for maternal behavior. So that fits the sort of the criteria of what prolactin might be doing. And as we've looked around at various circuits, we see examples of prolactin having very, very precise actions. So this slide is actually a late addition. But I just wanted to remind us that, we have this negative feedback pathway where prolactin is controlled by dopamine cells in the hypothalamus. They inhibit prolactin secretion. Prolactin controls its own secretion by stimulating those cells. And we've done lots of work over the years, and so have many others, to show that prolactin acts directly on those cells. It very rapidly increases their firing rate. You get changes in phosphorylation of STAT5. You get changes in phosphorylation of TH. There's a whole range of things that it's doing directly on those cells. So that's a very precise one neuron, one function kind of an action. But when we come to some of the other functions, it seems to be a much more broad network type of arrangement. So when Rosie did her work showing that prolactin in the preoptic area was critical for maternal behavior, she also did some work where we used the prolactin receptor Cree mice, injected them with a Cree-dependent virus. So you can see in this section over here, the green is tau GFP, so that's labeling all prolactin receptor containing cells. And then Rosie has injected the virus to label just on one side cells with M cherry, and then we could track the M cherry where it ended up in the brain. So what you can see over here is you've got roughly the medial preoptic area where we injected, but then the red dye turns up in a whole lot of other places. It goes to the BNST, the PVN, the ventromedial hypothalamus, the arcuate. There's a little bit down in the VTA. So actually, there's this action in the preoptic area could be acting throughout the brain. And we drew, in our paper, a circuit that looks something like this to illustrate the projections of the prolactin receptor containing cells. And the interesting thing that we observed is that wherever, in many of the places where we saw projections from the preoptic area, we also saw prolactin receptors expressed there in the cell bodies in that region. So it's kind of, if you go back and look at Bob's diagram, if we start with prolactin receptors in the preoptic area, we found that projections from there went pretty much throughout this network. But not only that, there were cells expressing prolactin receptor pretty much throughout this network. And so I've started now thinking about the way that prolactin works is to influence the overall network activity of some of these complex circuits that are driving things like maternal behavior. And when you think about it, that's an ideal thing for a hormone signaling to be doing. Neural signaling and neuropeptides are often, one-to-one communication that are defined by the anatomy, and you've got one cell talking to another cell. But if you want a system that the whole circuit needs to be tuned up or tuned down, then an endocrine signal is a good way to do that, because prolactin is circulating in the blood. It gets into the brain very rapidly, right throughout the brain, and can influence that circuit in how it functions. So that's kind of the end of my introduction, I guess. I've been thinking about why are there so many prolactin-responsive neurons and prolactin-responsive tissues, and trying to make some sense of the heterogeneity, the many, many different cells that express the prolactin receptor. And what I'm thinking is that this is an ideal sort of signaling system to allow broad-scale adaptive change. And pregnancy and lactation is a prime time where you see evidence of that broad scale of adaptive change. And I think that what prolactin is doing is coordinating all of these physiological changes that have to happen, and as well as the behavioral changes to allow the mothers to look after her pups. So, I actually have no timer here, so I have no idea what I'm doing, but yeah. I'll just keep talking until Zane pulls me off. So multiple body systems have to adapt, and so what I want to give you is a few examples of some research that starts to tackle some of these specific things. So if we accept that this list is not complete, but it does list quite a number of things that are happening in pregnancy and lactation, you will probably all agree, even though I tried to talk you out of it, that prolactin is involved in lactation. So we can give that one a tick. Hopefully you will be convinced by Bob's work and what we've added to that it's also involved in maternal behavior. So I'd like to start looking at a couple of others. The first one that I'd like to focus on is the change in fertility. I was just at the Kispeptin meeting before the satellite meeting and hearing a lot about Kispeptin, and so I really wanted to talk about this. But again, if you're a prolactin biologist, this is the obvious target, because if you're a clinician, for example, and they say, well, what happens if you have too much prolactin, hyperprolactinemia? What happens? The problem you have is infertility. And so it makes a lot of sense then that when you want to have infertility, such as during lactation, when you turn off the reproductive system, that prolactin is the optimal signal for it. But actually many, many people have done studies over the years, and they've identified, well, it looks like prolactin might be suppressing fertility, but also it looks like the suckling stimulus might, and in some conditions there's a role for prolactin and some not. And it's been a little bit unclear as to whether there is a specific role for prolactin as distinct from an inhibitory role from the suckling stimulus. So we thought we could contribute to this discussion. And the reason we thought we could add to that is because we now know so much more about the control of reproduction from all of the advances that my colleagues in this room have done understanding the role of Kispeptin. So this is work from Eleni Hackwell, who's a PhD student. She is just about to submit her thesis, and this just summarizes the model that we're looking at. So on the left-hand side is the pattern of LH pulses that we see in a non-pregnant female mouse. And what you can see is that in lactation, those pulses just completely stop. And you sometimes get them just starting, maybe one pulse starts up by about the second week of lactation, but they really stay pretty well suppressed until weaning, which happens on around about day 20, 21. So as I said, we've focused now on the role of kisspeptin because I don't have time to go through all of the background data, but I think you can take from this lovely diagram from Alan Herbison that these pulses, shown in red here, are predominantly driven by these arcuate kisspeptin neurons. So there's some element there that we refer to as the pulse generator, that these kisspeptin neurons are driving the pulsatile activity. So our question is, what happens to the activity of those neurons during pregnancy and lactation? To study this, we adopted the Gcamp fiber photometry method that was pioneered for the kisspeptin neurons by Alan's group, and I wanted to say special thanks to Jamie McQuillan, who was a research fellow working with Alan, who spent a lot of time training Eleni to do this work. For those of you who are not familiar with it, I'll just sort of touch on it briefly. It involves genetically encoded calcium sensor called a Gcamp 6, which essentially fluoresces more in the presence of intracellular calcium. So changes in intracellular calcium in the cell will lead to changes in fluorescence. And you can express that, in our case, in the kisspeptin neurons using the KISS1 Cree mice that Uli Bohm developed. And then you can insert a fiber optic cannula into the arcuate nucleus and essentially monitor changes in intracellular calcium in those cells. And Eleni gave me those slides, and she wanted me to add this picture because one of the key things in making this work is your mice have to be completely happy. Any amount of stress, and this system shuts down. So Eleni trains the animals for long periods of time, and they end up happily sitting on her hand while she collects a blood sample and has the photometry rig recording what's going on. I have to stay well clear because me bumbling around would just cause problems. And we have shown, replicating work that Alan's group has had pioneered, that these you get these amazing. I want to point at them, but this mouse is really touchy. These amazing sort of periods of activity that you can detect with fiber photometry, which is the population of kisspeptin neurons all activating at once. And that is very, very faithfully driving pulses of LH secretion in the mice. And this is critical to fertility, and there's been a whole conference on this coming up. So we then moved on to just look at what happens to the pulse generator. And basically the pulse generator shuts down as soon as pregnancy starts. That's not surprising. That's likely due to the high progesterone that is present in pregnancy. But what we see during lactation is absolutely no activity of the pulse generator at all. Just starting to kick in about, when the LH pulses start up, we start seeing activation again of the pulse generator. So as you would predict, the pulse generator is showing exactly what we would see based on what we'd already known from the LH levels. So we know that these kisspeptin cells express the prolactin receptor. It's now been confirmed by Brian and Giles on their new sequencing data. And this just illustrates that. You can see that in this case, the kisspeptin cells are labeled with red, and then the green is showing a functional response to prolactin. So this is an animal we've injected with prolactin and then looked at phosphostat 5. So all of the green cells are all of the prolactin responsive cells. So you can see that all of the red cells are showing this response, as well as a whole bunch of other cells. But all of the kisspeptin cells respond to prolactin. So the key experiment for us then was, well, what happens if we knock out the prolactin receptor from those kisspeptin cells? What happens to the lactational infertility? And really interestingly, the first thing we observed actually before doing anything else was just monitoring the cycles of these animals. And we found that basically in the animals that we knocked out the prolactin receptor from kisspeptin cells, well over half of them go straight back into an estrous cycle in the first week of lactation. We never ever see that in the control. So something dramatic has changed. It's a very, very long and complicated story as to why this response is so variable, but I actually think I know what it is. So the real aficionados can come and capture me later, and I'll talk them through. The way I'm interpreting this is that you need the prolactin receptor to maintain inhibition of the cycle. And sure enough, when we look at the activity of the kisspeptin neurons, this is really preliminary data because we were doing this in a lockdown, and actually getting people in the lab and animals at the same time was really difficult. But Elaine has done just 2 animals in this group now, but both of them have the same results, so that's encouraging. So on the left-hand side is our controls, and again, so the non-pregnant, this is the same animal. This is one of the beauties of this technique. It's the same animal following it right through. You can see in the virgin state, you're seeing these peaks. It's turned off there's a bit more noise than the previous example, but it's turned off, it stays turned off. It'll stay turned off for another 2 weeks. In the animals where we've knocked out prolactin receptors, here's the diestrous state. It turned off all the way through pregnancy, but then as lactation started, this system started to burst into action again, and you can see that, whoops, sorry, this mouse is too sensitive for me. You can see that the events of kisspeptin are happening again. So our interpretation of that is that prolactin is required for the suppression of the pulse generator function. Those animals are still suckling, they're still producing milk, they've got everything else about that lactation as normal, except the pulse generator has started again, and the cycle has started again. So that's, I would say, pretty convincing that prolactin has a role here in the suppression of the cycle. Okay, the next example I wanted to talk about were effects on metabolism and food intake. And we've actually done a whole lot of work on this over the years. And I'd say that the answer is still coming. We haven't really finished this role. So what I wanted to actually do talk about was a kind of a crazy result that came out of the study when we weren't intending it to happen. And this is work now done by Sharon Ladyman in the lab. She was basically setting up some animals in metabolic cages, and we had animals with prolactin receptors knocked out of various tissues, and we were going to run them through the metabolic cages and see what happened to their food intake, what happened to their body weight, all of this sort of stuff. And that is work that's ongoing. But what you'll notice in these particular cages is they had a running wheel. And to be honest, I didn't even think about it. It's just part of the kit, right? It just came out of the box. And so we left it in there. And it makes quite a big difference to what goes on in these experiments. But what Sharon noticed immediately when we were doing these experiments is that something crazy happens to running wheel activity in pregnancy. And that is, they stop. Mice, if you have a running wheel in their cage, they love it. So they will run on the wheel pretty much all the time. You can see the scale here is in kilometers per day, and your average female is cranking out 10K a day, a bit like me. Imagine with much smaller legs as well. So it's quite a bit of running. Every 4 days they run a bit more. It might have something to do with an estrous cycle. Anyway, they love it. And then as soon as they get pregnant, they stop. It goes about half what they ever did before, and then it fades away right throughout pregnancy. They go for a bit of a jog after giving birth. Lisa, where are you? How are you feeling after? Yeah, you didn't go for a jog, I bet. No. And then right through lactation, they don't do anything at all. And then you take the pups away, bang, they're on the running wheel. And when you look at a set of data like that, and you're me, you go, that's prolactin. And so, so sure enough, we did the experiments, and this is now published, so I'll just sort of jump to the punchline, which is, it was prolactin. When we knocked out prolactin receptors in a variety of ways, but this was, I think, the most clear-cut result, I guess, or the informative result is the word I'm looking for. So in this case, we did exactly the same manipulation that Rosie had done to characterize the role of prolactin receptors in the preoptic area. We used a virus to deliver Cree into the preoptic area, knocked out prolactin receptors from the preoptic area. And in this situation, they basically just carried on running right through the first half of pregnancy. They started to tail away when they got really big and fat, but even to right at the end when they've got, you know, full load of babies on, they're still running 2 or 3 times as much as the controls. So prolactin, for whatever reason, is massively changing the physical activity during pregnancy. We were thinking about this as like a metabolic conservation thing, you know, obviously for rodents and actually for females of any species, pregnancy is a marathon. You know, it's requires a huge amount of resources. So does lactation. Milk production is extremely metabolically demanding. So this might be just an adaptive response to conserve energy for something that's coming up. And if it is, that's quite a cool one because it happens immediately before the metabolic load comes later. So it's an adaptive change. But actually, I've got 1 or 2 other ideas about what might be causing this that I'll cover at the end. So to be honest, I haven't actually addressed the whole food intake side of it. That'll have to come another day. But at least I think what we can do is we can say there's another adaptive change that we didn't even know happened, but we can now add it to our list that is changing in response to prolactin. The final example that I want to give in the pregnancy part of the talk is another experiment that went badly wrong. And I kind of like to tell the honest, you know, I could give this story in a much more, we thought this and so we did that way. But I thought, particularly for the trainees, it's better to hear that actually sometimes you discover things by accident. And this is one of those stories. We were trying to do an experiment where we were back to our interest in the preoptic area. Obviously, we'd seen this effect on running wheel. We've seen this effect on maternal behavior. So what we thought we would do, wouldn't it be interesting if we used chemogenetics to activate the prolactin receptor containing cells in the preoptic area and see if we can induce some of these maternal adaptive changes. So that was the idea. And in fact, I think when we started, it was really about could we make males maternal by, or paternal by activating these parts of the brain. But anyway, so we put in a virus to drive the excitatory dread molecule in the preoptic area of the prolactin receptor containing Cree animals. And then we gave them CNO. And as I said, our idea was that we would expect that we were going to activate the neurons in that area and see a change in behavior. Can anyone guess what's going to happen here? Some of you have probably seen this story before because I have given a couple of talks where I've talked about it. What happened here is we induced massive hypothermia. And so the animals essentially just stopped. They certainly didn't show any parental behavior. They just basically stopped moving, curled up into a ball and did nothing. And when we picked them up, they were really, really cold. And so we went back and did some experiments where we put some telemetry recorders in them and did exactly the same experiment. And we showed this profound hypothermia. And, you know, what is going on here? So yeah, in hindsight, actually, this shouldn't have been a surprise. Hopefully none of you were surprised it was just me. Because actually, the preoptic area where we were mucking around with prolactin receptors is a very heterogeneous region. It's involved in a whole lot of homeostatic functions, one of which is the control of body temperature. And these data, which came out in this amazing paper from Catherine Delack's lab, this shows the sort of complexity of the cells that we're dealing with in the preoptic area. So they did single cell sequencing of the preoptic area, and they identified something like about 40 different cell types. And, you know, you could get so much out of this paper if you're reading it, but if you're me, your eye just jumps straight to this bit. So many of these cells have the prolactin receptor, and significant amounts of the prolactin receptor. And then if you read the paper, the actual word prolactin is used in the paper, which is exciting. And in talking about these neurons, the E13 subgroup, which obviously have quite a lot of prolactin receptor, I think they also had some leptin receptor. They've got a variety of other markers, but they appeared to represent the warm sensing neurons that Zach Knight's group had characterized previously. So there are actually a whole bunch of cell populations that are involved in thermoregulation in the preoptic area. And clearly what our results said is if you indiscriminately activate them, it causes hypothermia. And so we're very excited to actually try and work out what's going on with this. So we've looked at the warm sensing neurons. This is one example of the warm sensing neurons here. So in this, we've got staining for prolactin receptor is in pink, using our prolactin receptor Cree animals. And if you house the animals at 22 degrees, there's relatively little FOS, which is shown in the green. If you move the animals from 22 degrees to 38 degrees, massively activates FOS in this region. And so essentially the FOS labeling here, the green, is labeling what are thought of as warm sensing neurons. They're sensing the environmental temperature has got warm, they're activating, and in activating themselves, they start to initiate the response to warm, which is, to start cooling yourself off because the temperature outside is hot. And you can see that from this blow up that a large number of these cells that are expressing FOS in this region express the prolactin receptor. So somewhere in amongst the cells we've activated, we think that we have hit these cells. So it made us start to think about, well, what does prolactin do to these cells then? And I better show this slide in deference to poor old Tio, who should be here, but is holed up in a hotel room or something in Manchester with COVID. So Tio started to do some imaging, and this is very similar to the Gcamp that I talked about before. Here we're driving it with a prolactin receptor Cree, so it's in all the cells that have prolactin receptor, and we're looking at it in a brain slice, and I'll just start this video going. And so you can see that this has been sped up. You can kind of see the activity of the cells. And then we put prolactin on them, and you can see what has happened. This goes on for another few seconds, and then we'll put potassium chloride on to show what it looks like when all the cells are activated. They go bang. So clearly what happened when we gave prolactin there, it wasn't very impressive, right? Because basically most cells don't show an acute change to prolactin. That's a good thing, because the last thing you want when you're just getting a surge of prolactin, you're breastfeeding your baby, for example, is to suddenly go into hypothermia and keel over. So clearly that's not what prolactin does to these cells. When we look at these cells, most of them, we saw the occasional examples that were activated, showed acute activation in response to prolactin. We saw occasional cells that were inhibited by prolactin. The vast majority, we didn't see any acute effect. And so this, I guess, reflects the fact that we're dealing with a very heterogeneous population, and it'll be fun to work out what are the cells that are responding each of these ways. The bottom line is, if you look at a marker like phosphorylation of STAT5, in this exact same area, prolactin massively acts on all of these cells. So here's a blow-up area. You take an untreated animal, there's almost no STAT5, you give it prolactin, you see massive STAT5 throughout this area. So all of these cells are responding to prolactin. They're just not responding with an acute activation. So that CNO response that we saw is a kind of it was a flag that to go and look at temperature, but it's not physiology. So what is physiology? What happens? That's what I asked the question now. So clearly prolactin can influence the temperature sensing circuits. Why would that have to happen? What happens to those circuits in pregnancy? Probably all of you who are in the room who have been pregnant could tell me a story about this, but I didn't know. So we went looking, and again, this is now just wild-type animals. We've put a telemetry device into them and monitored body temperature throughout pregnancy and lactation. And it's really quite amazing. So normally you have this kind of circadian rhythm of body temperature. As soon as the animals are pregnant, the circadian rhythm goes away and body temperature drifts up a little bit. I suspect that's got something to do with progesterone. The very high levels of progesterone, it's going up and staying up. And you can see that as you go through pregnancy, it edges up a bit higher. But then in late pregnancy, it drops back down a little bit. And that's really interesting, because at that point, the metabolic load on the mother is highest. She's got developing babies that are making their own body heat. She's got generating all of this metabolic heat. She's got the mammary gland starting to be functioning. And, so there's a huge metabolic load, but her body temperature is coming down. And so I think this is an adaptive response, because high body temperature is actually bad for the baby development. So the mother is having systems, adapting those systems to lose temperature. And so we're thinking that maybe this is what prolactin is doing. So we've started to look at that. And this is, again, using the exact same model that I've been showing you for other things, where we've knocked out prolactin receptors in that preoptic area and then monitored their body temperature through pregnancy. The black lines here are the controls. You can see they're going through that same cycle of edging up and then down in late pregnancy, dropping back down. But in the absence of prolactin receptors in the brain, they don't show that adaptive response. So the body temperature, as shown in red here, just edges up and up and up. Unfortunately, with this model, as I told you right at the beginning, these animals don't look after their pups, so they abandon ship, and so we can't really follow what happens in lactation in these animals. But this might be one of the reasons why they abandon ship, because they're not coping with the temperature changes. Anyway, to try to follow this better, we decided we needed to use a different model, and we went instead to, we knew all of these neurons were glutamatergic from all the work that's been done before, so we used a V-GLUT-CRE to knock out the prolactin receptors out of all glutamate cells. So it's not a perfect experiment. This is all glutamate cells, not just the ones in the preoptic area. But we knew from our previous work that the V-GLUT-CRE prolactin receptor knockouts are able to lactate. They look after their babies just fine. So again, we thought we would go and see what happens to the pregnancies in these animals. But because of that effect of the body temperature rising in the knockouts, we thought maybe it would be interesting if we gave them a little bit of a thermal challenge. So in this case, we've got some animals that are housed at 22 degrees, which is our normal temperature of our animal rooms, and some that are at 30 degrees. 30 degrees is kind of thermoneutral for a mouse in a mouse room, and what that means is they have to spend no energy to lose temperature or to maintain their body temperature. But if they want to lose body temperature, it's quite a lot harder because the environment is warmer. And you can see that even our control animals at 30 degrees, there's quite a number here where actually the pups didn't make it. So the pregnancy failed and the mothers abandoned their pups. But on average, there was no difference in our controls. But when we looked at our knockouts, again, it was catastrophic. At 20 to 22 degrees, they were fine, absolutely fine. They could look after the pups. They were obviously able to lose enough of their body temperature. Everything was fine. But at 30 degrees, they were saying they wanted none of it. And I think this is really a really profound result, mostly because it was completely unexpected. So just really quickly, thinking about circuits again now, we've been focused on the preoptic area, but it's actually the thermal regulatory circuits are well described. And this is just from a news article from Cliff Saper in Nature, and he was kind of mapping out some of the thermoregulatory circuits. And we, of course, have been interested in this preoptic area that has prolactin receptors. But actually, there are prolactin receptors in the dorsomedial hypothalamus. There are prolactin receptors in the raphe pallidus. There are prolactin receptors in other places that might also contribute: dorsal root ganglion, brown adipose tissue, skin, sweat glands. Why would there be prolactin receptors in sweat glands? Thermoregulation. So I guess this illustrates how we're going. I'm hoping there's about 5 minutes left. Anyone got a, yeah, okay. So sort of to summarize all that, basically what we've got is the idea is that prolactin, placental lactogen, acting throughout the brain and the body for that matter, potentially on completely different cell types, modulating completely different functions: physical activity, maternal behavior, thermoregulation I showed you. It's acting in other brain areas, so I showed you the lactational diestrus of the Pepto neurons in the arcuate nucleus. We've recently published that prolactin acts in the ventromedial hypothalamus to affect maternal aggression. It affects the stress response. Out in the body, of course, it affects the mammary gland. Pancreas, it's required for the beta cell changes that happen during pregnancy. All of these things, and the old me would have looked at that and said, look at all these functions of prolactin. So many different functions. How cool is that? But actually, I think it's even cooler, and I now think of it as that's not a whole lot of different functions. That's one function, which is maternal adaptation to pregnancy. And that's what I think this hormone is doing. Okay. So I've shot myself in the foot here, haven't I? If you take this graph and you take out the words animals that lactate and you say, okay, how many of these animals go through pregnancy and have placentas and all that sort of stuff? It's pretty much the same sort of group. So what else are the rest of these animals doing with their prolactin? Well, one thing we have looked at is males. And this is a photo that I took on safari with Mike Ludwig and Alan Herbison when we're on our way to a serious endocrinology conference. And it was just an amazing sight. This is in Etosha National Park in Namibia, and we just, we pulled up alongside these ostriches, and they couldn't care less about us because they were doing parental behavior for birds, right? When it's hot and you're in the desert and you've got thousands of little baby ostriches, whatever they're called, there's no shade, so you have to be the shade. So that's what you do, parental behavior. And you can see mum is doing it on the left and dad is doing it on the right. They're both doing it. So we started thinking, obviously I'm possibly the only person in the world who looked at this amazing sight and said, oh my God, that's prolactin. So we thought about prolactin, what's it doing in the male? So going back to mice now, prolactin receptor distribution in the male brain is actually almost exactly the same as the female brain. In the preoptic area, where this maternal behavior thing happens in the females, the distribution of prolactin receptors in the male is kind of similar. There's a few less neurons, but basically the distribution of the cells is the same. So we've been interested in asking the question whether prolactin has an equivalent parental behavior role in the males. And this is a diagram again from Catherine Delac's lab, just sort of describing what happens with paternal behavior in males. Because your average male mouse, if he's a virgin male mouse, he really doesn't like pups at all. And if he sees pups, he'll attack them and sometimes kill them. So we call this infanticide. But they have this incredible behavioral change where if the male has the opportunity to mate with a female, and it requires the mating stimulus and ejaculation, if he's done that, 2 weeks later, his behavior completely changes, and if he comes across a pup, he will pick it up and look after it and nurse it. And so that behavioral switch is phenomenal, but it makes them a really interesting model to think about paternal behavior in mice. I mean, the interesting thing about that is the timing of that change is almost perfectly aligned where if there are pups around, they could be his. But he'll look after anyone's pups at that stage. They don't have to be his. So it's just this behavioral switch that somehow is happening in the mice. And we were interested in whether prolactin was involved in that. And I'll just show you this video, hopefully. Yeah. So now we've got basically on the left-hand side control mice, and on the right-hand side knockout mice, prolactin receptor knockouts. We basically knocked out prolactin receptors from as much of the brain as we could with a CAM kinase Cre. And we put the baby pups in there, and you can see that as soon as we put the pups in, they know that they're there. The guy on the right knows they're there. I mean, he keeps on coming out and checking them out. The one on the left is behaving just like a female. He's picked up one, taken it to the nest, he's putting it back, going to get another one. That's normal paternal behavior for an sexually experienced male. The one on the right just doesn't seem to know what to do. He knows they're there, but he never retrieves them, never. Just leaves them there in that little pile. And so this is just the data showing that. You can just focus on what's on the right. So the pups retrieved, the controls pretty much all retrieved them, the knockouts never did. So prolactin seems to be required for parental behavior in mice. We were interested as to thinking about when would this happen? Is it the mating response? Because actually mating induces prolactin secretion. And so one of the things we looked at is we took some animals, we allowed them to mate so that they had this little ejaculation-assisted rise in prolactin. But we had another group of animals that we treated with bromocriptine, so they didn't show that rise in prolactin. I was pretty convinced this was going to screw with their behavior, but actually what happened is nothing. So here are the virgin animals, they attack pups. Here are all the mated animals, whether they were control or bromocriptine treated or bromocriptine treated given prolactin, they never attack the pups. So that transition from being aggressive to non-aggressive doesn't require prolactin. Moreover, they all retrieved the pups as well. So this rise in prolactin that occurs at the time of mating doesn't seem to be involved in the behavior. On the other hand, prolactin at the time of seeing the pups does seem to be involved. So now on the left-hand side, there's no actual acute change in prolactin when the males see the pups. Prolactin doesn't change, or you can see this is just kind of a before and after the pups on the left, and some of them go down and some of them go up, and there's net no change. And it's actually really low all the time. But what we did is treat a bunch of the animals with bromocriptine so that their prolactin levels went down prior to seeing the pups. And in that case, they failed to retrieve. So when the bromocriptine treated animals failed to retrieve, if we gave them some prolactin back, just in case it was something crazy that the dopamine agonist was doing, we gave them prolactin back, and that reversed the behavior back. So they actually, just that low level of prolactin that is normally present in a male is enough to induce this parental behavior. And we've actually gone on to a really a fabulous collaboration with Christian Broberger in Sweden, and shown that this prolactin action is in the preoptic area. And you know, he's characterized this amazing difference between rats and mice into whether they'll show behavior or not based on the activity of their dopamine and prolactin system. I had a slide about that, but the system didn't want to show it, so it's out. Okay, so I'm just going to finish a couple more slides thinking about what about other animals. So I'm from New Zealand, as you probably worked out from my accent. So I thought I'd better show a kiwi. A kiwi is not a little green fruit with brown skin. A kiwi is a little brown bird about the size of a large chicken. The female lays these. This looks like it's Photoshop, right? This is an actual kiwi egg. They lay one egg, and when they're through with that, they are not remotely interested in that egg. The female will lay that egg, and she's off, and the male does all of the incubation of the egg. So male parental behavior, birds' male parental behavior is a common thing. We're not allowed to do research on kiwi, but actually Christina, who in her previous life has done a whole lot of work looking at parental behavior in birds and showing that prolactin has a role both in males and females in maintaining this parental behavior in birds. So I can sort of think about, well, I'll go through my final conclusions. I think what I hopefully have shown you is that prolactin is not only a lactation hormone. I don't want to diminish the role of prolactin in lactation, because 100% is required for lactation. In the absence of prolactin, there is no lactation. So it is definitely prolactin, but it does other things. And I think the high levels of prolactin and placental lactation during pregnancy are required for this adaptive changes that are happening. And you can kind of think of this as like the classic Pleiotrophic gene. You're having multiple different effects to you know, that seemingly unrelated but actually have a common goal. And I'm really excited by the idea that they're coordinating the physiological changes as well as the behavioral changes. So you're getting things like the thermoregulation, physical activity, glucose homeostasis. Those are all changing, and prolactin's contributing to that at the same time as the behavior and the maternal behavior and the nursing associated with lactation. So, and I think the way to think about this is that this is like the ancestral role of prolactin. So you can actually have examples. I gave you some examples from birds, but you can also see examples from fish where prolactin seems to promote the parenting interactions between the parent and their eggs or their offspring. And in that context, you could think of lactation as just the latest addition to a parental repertoire that was added when the mammals came along. And perhaps this is also the reason, you know, I study prolactin, and I must have been asked the question 1,000 times, well, what does prolactin do in the males? And I've always kind of waved my hands and said, well, probably something. And what I think it does is almost exactly the same as the females. It promotes parental interactions. Almost certainly this is not all that prolactin does, because there'll be other times when prolactin is elevated, such as during stress, and it might have another set of roles. But I think this is a good construct of how to think about it. Okay, so I'll finish with the important slide of thanking everyone who did it. I was very embarrassed when I put my talk together that I don't have a photo of my lab since before COVID. And I think the reason for that is I'm so unused to traveling that, you know, I was packing and going away, and I didn't organize the lab to take a photo. But anyway, the key people are here. I'd like to thank, if I can get my pointer working, it's not going to work. So the people that I've mentioned as I've gone through, we've got Eleni, who did the work with the photometry, Rosie Brown, who was in my lab for a long period of time. She now has her own position in the physiology department at Otago, doing work on parental behaviour. Sharon over the back there, who did all the metabolic stuff in the running wheel. Christina, who was involved in the male side of it. Tio and Joe, actually, who both are attempting to go to this conference and got COVID on the way here. Tio, of course, doing the imaging stuff. And thank the people who funded the work. And just in case you're interested, we do actually have a position available in the lab working with Rosie in particular. So anyone who might be intrigued by the roles of prolactin, come and see me afterwards. All right, thank you.